Okay, so today I'll be taking a topic which is advanced clinical trial design, how the studies will be designed and what are the different types of studies designing we have got previously and how it has been implemented during our clinical trial design. So my first three designs I'll be discussing today is the first one is adaptive design, the second one is master protocol, and the third one is synthetic arm. So when you carefully see, the adaptive design is the one which is at most used now currently in industry. And master protocol was very much useful during COVID time, where we were testing multiple drugs for one purpose, which was COVID, and synthetic arm, where we are using the real world data or studies to conduct the current treatment efficacy or safety. So this is about adaptive design. So let's first understand what is adaptive design and how it is being impacted now. So during earlier times, we used to design a clinical trial in such a way that we design a protocol and we go and enroll the subjects, we collect the data and we conduct the clinical trial and then we analyze the data. So this was a straightforward design, which is a fixed design. So there was nothing which was being changed during the conduct of clinical trial. So this design was called as fixed design, and there was so much of challenges in this design. So at any point in time, if you wanted to change the design, it was not happening or it was not possible in fixed design because you design, conduct and analyze the data. But while coming to one of the most important aspect, which is adaptive design. So you design a clinical trial protocol and you conduct a trial. And during a conduct of trial, you stop a trial and you collect the data. You collect the data, you analyze the data, and you come up saying that whether I'm going in a right direction or not. And you give a new adaptation or new things to be adapted, and you review that adaptation. And one more time, you conduct the clinical trial from there. And then you go ahead with analysis of data. Analysis of data. So this is something which is called as adaptive design. So you design and then you conduct and then you implement the designing. You implement the designing, you review the designing updates, and then you implement it during the conduct of clinical trial. And finally, you analyze the data. You analyze the data to make sure that the design is adapted correctly. So now the adaptation means you keep on changing the trial during the conduct of clinical trial. So now with an example, I will see the fixed dose conventional method. So a patient is taking, a group of patients are taking treatment A, will go for interim analysis. You stop a trial and you go and do analysis, and then you will go for final analysis. And this final analysis, how many subjects you have thought, those many subjects only we will analyze. We will not change the subject size or will not change anything of this particular aspect. In the same way, in the one more group in conventional fixed design, treatment B we will give, and then we do interim analysis where during the conduct of trial you stop it, you analyze the data, and you go for a planned sample size which was earlier decided, and you don't adapt any of the changes in this. So, but coming with the next design, which is very interesting, designing with SSR, sample size reassessment. So now we keep on changing our method and adapting new things during the conduct of clinical trial, and you modify the number of subjects to be taken into the clinical trials. To be taken into the clinical trials. Now, what is mean by the sample size reassessment is you give one group of subjects treatment A and you go for interim analysis and you keep on increasing the number of subjects, which is seen here, which is adapted sample size. During interim analysis, you came to know that this number of subjects will not lead to a right result or right outcome. You change the number of subjects, you increase the number of subjects, and you adapted this design for a conduct of clinical trial after interim analysis, understanding that the number of subjects might give more information or more right output. In the same way, for treatment B also, designing with sample size reassessments, you do interim analysis and you keep on increasing the number of subjects. And of the next one, designing by adaptive enrichment. So now first group, you give a treatment A, and then you go for interim analysis and you find out the subjects who gives the good results. A particular subjects who are giving wonderful result, which is marked in green color. You keep on adding more of green colored subjects, which are being highlighted here, and the existing subjects, you will continue it here, so that you will get more data for the good results to be given. So now this design is called as design by adaptive enrichment. So now in this particular way that we are going to do adaptive design, which comes into picture that during the conduct of clinical trial, we keep on changing our clinical trials subjects or the route of administration or the efficacies or the safeties or any kind of aspects you wanted to put it into your clinical trials. Then this is something which is called as your adaptive design. And this adaptive design is particularly used during combination of phase 1 and 2 or phase 2 or phase 3. So now when you want to do phase 2 and 3 together, then you go for adaptive design, where you start with one particular design, but while conducting, you will adapt for new things which are happening in clinical trial. And this is a very, very clear that the intent, the need of the adaptive design is to use the data which is collected at earlier stage, so that we can adapt new designs moving forward. So we want to collect the more information during the initial phases of conduct, and we put a checkpoint for interim analysis, and we change or modify the design of the clinical trial to meet our expected outcome. So now this is something which is your adaptive design, which is furtherly used for conduct of clinical trial. Now, nowhere we are going and saying that, okay, you design a trial and you conduct and you analyze. We say that you design it, you conduct it at initial stages, only collect the information, data, and then you might change dosing regimen, you might change the sample size, which I have shown the example, you might change the subpopulation group, you might change the arms also. So now this is something which is your study design adaptive method, which is furtherly used for making sure that our trial is meeting the perfect criteria. So as per FDA, how the adaptive design is being defined. So a clinical trial design that allows prospective planned modification. Now, during conduct of trial, we are modifying the design. Modification to one or more aspects of design based on the accumulating data from the subject of clinical trial. So now the initial accumulation of data will be further used for properly changing the design, either it is dosing regimen or arms or sample size or study population. But there is a big difficult challenge in this adaptive design, that what we have earlier designed, it will be very, very flexible and it will be very agile, that the design gets changed and the impact of the data which is being collected is more problem. And particularly when you are randomizing the subjects with IVX or IVRS or IWRS, so it's very difficult to change the dosing regimen, because if a subject is randomized to a treatment A, during design you wanted to make it as treatment A and B, it's very difficult because of a limitation of system. But yes, this design is to adapt the new changes with the existing data to make sure that we are moving into the right pace or right format. So now, if you carefully see, I have given one more example of the interim analysis. So the first one is conventional trial, which is fixed design, treatment 1, and you go and original planned sample size, and standard of care, which standard of care means the already existing the subjects what are being taken is called as standard of care. And this subjects, one more time, original planned sample size we will take and we will analyze. So now next design is adaptive clinical trial sample size reassessment. So you start with treatment 1 and standard of care, but you end up increasing the number of subjects so that you get the right results, and you adapted this design after your interim analysis. And now the next one, adaptive clinical trial with response adaptive randomization, wherein based on the response of the subjects, you will keep on increasing the number of subjects which is being given here. And last one is adaptive clinical trial with enrichment design, where the output of the subjects who are being given this outcome will be increase the number of subjects based on interim analysis. So now adaptive designs, first of all, we will do background review. How can we implement it in a proper way? And you go ahead with trial simulations. And if trial simulations is ready and you finalize a protocol, if not, you discuss and further you go for trial simulations for this prospective area. And when you go for implementation of adaptive trial design, you first design a trial and you burn in period or run in period. You make sure that your subjects are under controlled environment and you go and collect the interim analysis report. From interim analysis report, you keep on changing your design. So now sample size, whether it is reached, if it is yes, you go for final analysis. If it is no, we will ask whether it is ready to terminate. If it is ready to terminate, you stop the trial, you go for analysis. If it is no, we'll ask whether the adaptive decision criteria are met or not. If it is yes, we adapt the trial design. If it is no, no adaptation. Either it is adaptation or no adaptation, we continue the trial and we go for final analysis. So now this is how you implement an adaptive design in real-time scenario, and this will make sure that the use of data collected in earlier stages will give more answers for you for further study analysis. Now, these are the advantage. The probability of success will be more because you change your design during conduct of clinical trial. Maximum information you can use it because at the initial stages only you are collecting and putting in interim analysis and you're using it for adaptive designs, and shortens the development timelines, which is reducing of your time for putting this clinical trial. Overall development cost, money will be reduced because you keep on changing very flexibly for the design, reduces the risk for the study volunteers and as well as sponsors, subjects, and sponsors will not have more risk to conduct clinical trial, and maximize the probability of success with the regulators and also the players. So this is one more very important one, the master protocol, or the one protocol which will use for multiple purposes. Now, when we had got COVID, so many people started asking question that which treatment might work, which treatment might not work. So now in this scenario, there was all of the situations where A drug can work, B drug can work, C drug can work. So now we really don't know which drug can work, A drug or B drug or C drug. So altogether, different drugs, can it be tested in one particular study? So this was the question which was being asked particularly during COVID time. And then we came up with an answer is, yes, we can do it with a prospective area of master protocol. So now master protocol is the design that includes multiple subgroups. You will have multiple subgroups. Group A will take one treatment, group B will take one treatment, group C will take one treatment. With the patients having same diseases or different diseases. In COVID situation, it is the same disease. But in other diseases, you can have multiple diseases, but the same drug also, where one or multiple drugs can be treated. So now we need to understand that it can be one drug tested in one disease or one drug can be tested in multiple diseases. So multiple diseases, one drug, or one disease for multiple drugs. So there are so much of challenges in this master protocol where you can test for different diseases, different conditions, or different indication, one drug. In the similar manner, you can use in an opposite way also, multiple drugs can be tested for multiple diseases or one disease. So this is one thing which is called as master protocol. Earlier days, we used to say, okay, one drug, it will treat one disease, let's do it. So, but now it's in the opposite way, that why are we testing only one drug? If we have 5 drugs, can't we make it as 5 subgroups in one study, in one master protocol, and give it to the patients? Now, this is something which is called as your master protocol. Master protocol design is intended to simultaneously evaluate more than one drug with more than one disease with overall trial structure. So now single infrastructure trial design protocol can be used to evaluate the multiple drugs working in multiple disease population or multiple sub-studies so that it speeds up the drug development or clinical trial approval process. So here we need to understand that earlier days we used to say, okay, one drug can be tested in only one disease. But now we are saying one drug can be used for multiple diseases. And here we are saying multiple drugs can be used for one disease. So both things are possible. So one drug, multiple diseases, multiple drug, one disease. Both things are possible so that we can test multiple aspects, multiple situations, so that our drug approval process becomes very faster. So now during this master protocol types, the first design which I'll be discussing is your basket design. So now when you call it as basket, so the right hand picture is being given as basket design, which is you have one drug, which is cancer drug A, within a very, very clear example that one drug is there. So which is your cancer drug A. So assume that the drug name is cancer drug A. But this drug, we are testing in multiple diseases. We really don't know. This drug could have been having efficacy or indication or safety on multiple diseases. So now here, very simple example, one drug, multiple indications. You are testing one drug, multiple indications. So now one drug, you are giving it to one group who are breast cancer, pancreatic cancer, lung cancer, and colon cancer. At the end of the study, we might lead or we might come up saying that this drug might have efficacy on drug A or disease A or disease B or disease C, wherein it is very, very good aspect for conducting of the clinical trial. So this is something which is called as master protocol, which is basket design. One drug, multiple indication. A protocol which is employed in single therapeutic in multiple patient subgroups divided into parallel sub-studies. So now subgroups will have the expanded to achieve the clinical significance. So one drug, multiple indication is called as basket design. Now the opposite type is umbrella design. So you have multiple drugs, but you have only one indication. So multiple IPs are being tested for one indication. Now drug A, drug B, drug A plus B and control arm, you are testing all these multiple drugs for only one condition, and that condition is your lung cancer. A protocol with one or a protocol with more than one targeted therapy for a single disease, only for one disease, are divided into multiple parallel treatment arms. Now this is one arm, this is one arm, and this is one arm, and this is one arm. So during this multiple arms, we are testing it with multiple drugs, and for particular drug which is or particular disease which is your lung cancer. Now to be very clear cut short in terms of how this design is being done, umbrella design, multiple drugs are being used for one indication. So now this is what you call it as umbrella design. So now coming back to one of the most interesting and most spoken about this design is decentralized trial. Now every patient, give me 1 minute guys, give me 1 minute. Yeah, every patient has to travel to site. Is this needed or not? So answer is might not be needed. Now every patient home has become one site. So now, or else you can call it as siteless or complete virtual visit. So now this something which is called as decentralized clinical trial. So earlier days, a patient used to come to site, he used to give assessment and he used to collect the data, and then we used to close the site. But now the current design has been happening something like this, that there is no need of patient coming to the site and giving the information. So in this below example, I'll show you how it happens. Patient is at home and patient drugs are shipped directly to patient home. So patient is getting the drugs to patient home, and first he will take dosing visit. which is home visit. He is not coming. And before this first visit, we are taking telemedicine. By use of the video calls and all those things, the doctor and patient is getting in touch, and he's taking a drug. And he's also taking the labs, vital signs, x-rays, and consent. And he's also taking COAs and your PROs. So the clinical outcome assessment and the patient reported outcome, both data he's taking at the home visit. And the next visit also, it's a virtual visit, which patient does not need to come. And the patient will directly go and give vital signs. And the next visit is also home visit, where patient does not need to go to any site or any virtual site. He will take the vitals and as well as lab assessment, and also the COAs, clinical outcome assessments. And visit 3 is a virtual visit where he will just take the vital signs. And last visit is home visit, exit. So now it's a complete decentralized clinical trial where patient does not need to travel and reduce the time, money, everything, effort for the patient and money required for conducting clinical trial at the site. But the challenges here is we need to use digital technology in clinical trials or decentralized trials. Either it is telemedicine or collecting the data from different parts of subject, or variety of data will be coming, external vendor data or X data or Y data. And here SAEs also has to be collected with respect to decentralized trial. In a simple nutshell, patient does not need to go to site to conduct a trial, and every patient home is the site. Now here, if it is site, if you see carefully, now patient does not need to go here. Patient will go to a lab facility which is very nearer. You give the sample here, and the samples are being pushed into the site or your virtual site. In the same way, patient home becomes every site for conduct of clinical trial. So now there are so many new things happening in decentralized clinical trial, like e-source data or COA data or devices, wearables, sensors, where the investigator can remotely answer the questions which are being raised by the subject by the use of the telemedicine or by the use of technology. Not needed that patient has to travel every time to take a drug or medication or to give further assessment. During this decentralized clinical trial, we use a lot of technology. Now, there is a small one thing that it can be a hybrid mode, that some of the visits can be completely home visits and some of the visits can be site visits. Now, say for example, if a trial contains 100 visits for one subject, now nearly 80 visits you can say home visit. And another 20 visits, you can say that, okay, you can go for virtual visits. And this is something which is called as decentralized trials. Nutshell, it is referred as siteless or virtual study model. This design place patients at the center with an aim to limit or eliminate the need of the patient to travel from investigator site. So this is something which is you have for decentralized clinical trial. And coming back to platform design, so already I have spoken about the trial design, about trial design to have the basket and as well as umbrella design. But now combination of basket and umbrella design is your platform design. So you have platform design wherein multiple drugs, multiple diseases. So now in basket design, one drug, multiple indications. In umbrella design, multiple indications, multiple drugs. But now here, multiple drugs and multiple indications. Now this is combination of basket and umbrella design. So now the platform design where multiple IPs are being tested, across multiple indications. And this complexity of both umbrella and basket design. So now here you have multiple drugs, might be A drug, B drug, C drug, multiple indications, 1 disease, 2 disease, 3 disease. So you are taking in one trial wherein multiple diseases are being taken and multiple drugs are being taken. So this is something which is called as the platform design. And this is more interesting, that synthetic arms, and this is emerging study design where one or more arms are being replaced by previously collected real world studies or clinical studies. Now, what we are trying to do is, we have already conducted so much of clinical trials on particular drug or indication, and we have already collected so much of data from the real world. So now the problem is, why are we want to take one more time new group of subjects to test the same drug when you have already existing data which is already available, and can't we use it as synthetic arm? And the drug which you wanted to test, you will create a new group and compared with already existing data which is there in the market. So now synthetic arms is emerging design which are replaced with previously collected data. So already we have data. So now say, for example, if I have here candidates with historic of eligibility criteria, you have eligibility criteria 1, 2, 3, those subjects meet 1, 2, 3, will go into my trial. So we collect the already existing information and we dot it. So the dots which are dark brown are meeting my criteria and will directly go into trial, and you will not test here anything, because already that subjects have completed their studies and we already have that data. You just test the new drug, new control group against already exposed subjects or real world data. The secondary data is available and you have to assess the current arm, current drug, which avoids the unnecessary exposure of the patient to drug. But this design has some challenges, that already the subjects have taken the drug and those coding is very difficult, and they might not give proper protocol deviations when you have collected the previously data to conduct the clinical trial. And this is something which is called as your synthetic arm trial design and quality by design. So you have 4 words: you plan a design, and you do implement the risk management strategies, that before you start a trial, you identify what risks might get into clinical trial protocol. And you try to check, monitor to prevent the risks, and you act to remediation and learning for the risks. So you plan, you do, you check, and you act. And these 4 things are most important point in assessing your quality by design or quality by design clinical trial, and why these new designs are very much useful. The factors, what are the factors for clinical trial data or critical important data? So most importantly, the design of trial. You need to understand how this is being designed, what are the changes it's going to be happening, and feasibility of site, and the patient safety, and how are we going to conduct the clinical trial, and study reporting, the reporting of study data, and last one, third party engagement or third party data. With this, I have came to an end. So I'll repeat all the aspects in nutshell so that we understand all these things. Adaptive design, which is we collect the preliminary information to change the design and implement the design, adapt the design, review it, and conduct once again during the clinical trial. This is about adaptive design. Master protocol, which comes into umbrella, basket, or platform designs. One drug, multiple diseases, and multiple diseases, multiple drugs. Master protocol, one drug being given for multiple indications, which is basket design. Here, one in disease, multiple drugs. Decentralized trial, the conduct of clinical trial without patient traveling to the center, wherein it happens with every patient home has become the site. Platform design, multiple drugs, multiple indications. synthetic arm, already existing studies, clinical studies or real world studies we are using for the current arm, which is the synthetic arm, the previous exposed data can be taken. Quality by design, plan, do, check and act. And these are critical to quality CTQ factors for data into clinical trials.